Morning, everyone. So this is all new for us. This is my first virtual mini conference, and we have been running early a little bit because the breaks were not in the webinar. So this is all new for us. This is my first virtual. Now I have a huge echo, and we have been running early a little bit because the breaks were not in the webinar. This is all new for us. This is my first virtual. I think you need to mute your microphones, please. And we will now go ahead with questions. You can post questions online. And we will read them out to you. We will now go ahead with questions. You can post questions online. And we will read them out to you. Okay, sorry. Didn't realize that was me causing the echo. So yeah, as I said, it's all new a little bit for us. I also don't know how many are here now, because officially in the program we are starting at 11 with the discussion. But since many of you are listening now, we can go ahead and take questions. And Lucy, you already have a question from the live webinar.
We do. Good morning, everyone. So I will start with the first question from Arda. Thank you for your question. So they say, we have faced some news about unreplicable fMRI research, and there's a debate ensuing for fMRI measurements doing the same task in the same participants. And they say that they think that the situation can have an impact on brain mapping and modeling, etc. And so the question is a combination of EEG and fMRI sufficient to solve this issue?
Is that a question for me then?
I think it is, yeah.
Right. I guess that gets us a step closer, at least in my mind, especially in an EEG-informed fMRI analysis, where effectively each individual subject, the way we analyze the EEG data, each individual subject, its own sort of replication unit. So we cross-validate our results. We use these multivariate methods to try and extract trial-by-trial variability from the data that's as reliable as it can be, and do so better than, say, the more conventional standard ERP analysis. So in that respect, we can. I hear something in the background to you, which is actually quite distracting. Could that be the host, or? So right, just to carry on, yeah, and then using that as predictors for the fMRI, we're hoping that we can tap really into this endogenous variability and hopefully see activation maps that are hopefully more reliable. But then again, the best solution to all this, I would imagine, is to try and replicate the work both within the group, but also across groups, which is, I guess, why it's important to share data openly, and we've done that for most of our EEG fMRI data sets, which are actually available on Open Europe.
If I may add to this, I think this symposium shows quite nicely how the challenge is to, the challenge to replicate, that was the question, is met by replicating across species, which of course is a very, very demanding task. It's not only replicating across different different subjects and different methods, but also, like Matthew has shown, using the figure ground paradigm to replicate that between monkeys, humans, and rodents, is of course a total different scale. So we don't need to talk about numbers and replication crisis if we are able to find corresponding signals in different species.
Yeah, it was great. It was great to see in Andrew's talk that you were able to see the orientation tuning profile in the superficial and deep layers, which really replicated what many people have shown in the laminar studies. So that's a clear case where there was a clear target from the animal work, which is completely replicated in the human paradigm.
Yeah, exactly. We were really excited to be able to actually see that level of detail, which came from specifically trying to aim to replicate something using a technique that hadn't really been applied previously.
May I also add to that? There was recently a paper, maybe that refers also to that, which showed that in the same individual, kind of the same task was not reproducible well with fMRI, made quite some noise. And so of course, what you are all saying now, I fully agree, of course. And it shows how in what kind of high spatial resolution we can even find a validation that we measure the same thing. But I think here it's a bit different. This paper is a bit strange in my opinion, because it's not sorting out well alignment issues. It's not sorting out well the tasks might change of the same person over some time. So if you use more higher level cognitive tasks, I mean, I'm planning actually, if I would have the time, to publish data from the same brains over 20 years, showing how super consistent they are, not even going into laminar fMRI, right? So therefore, even at the more larger scale, we do not see that at all, what they claim in that paper. And I think it depends on so many factors they might not control. Like people, if I do imagery 20 years later, I might do it differently than I did it at the time. And that gives different patterns. That doesn't mean we measure something wrong, right? So therefore, fMRI has its problems, sure. But I think the recent critique that fMRI is scary because nothing can be reproduced, which is basically a sentence in that paper, just be a bit more straight, is really a very bad signal out to the community, given what is shown in this nice mini symposium. Yeah.
I would absolutely agree with that. I mean, in a lot of the talks today, Uri's talk, we saw replicability across individual subjects actually being analyzed together. In Marius's talk, you're using multiple modalities. Your talk and my talk, we're also looking at individuals and how very, very small effects or specific effects that we have hypotheses about were replicable across individuals. And then, of course, looking at across species to be able to see these effects reproduce is pretty incredible already.
Rainer, I have a question for you. What does tiramisu mean, and how did you come up with a name for that?
Well, the name is. I know, of course, you have done very nice work in a very similar direction with your Cerebrum is called, right? Your name is nice, sounds to a sci-fi movie, actually. And what we didn't know that you also do this for 70, honestly. I have seen this recently that you did that. So we wanted to do something which is especially focused on the issues we have with segmenting high field data, because there was already also before your work, others doing deep learning segmentation. So we wanted to have also a network which has some properties which I think are differentiating also our network from yours, because we basically can train that network with about 5 brains to a generalization performance, which is amazing. And that's because of the architecture of that network. Because this tiramisu network is called because it has this very sophisticated sets of dense layer in the encoding decoding phase. So it's like a very sophisticated UNet, which is basically the core structure. And as in your network, it's not possible because of the whole brain approach to do some of the more advanced current new developments in deep learning. And we basically put them all in. So ours is basically, the technology is just out from end of last year, basically, or mid last year. And it's basically allowing you to have more generalization capability from less training data by using these sophisticated dense layers. And the architecture we used has been called tiramisu net. It's an extension of 3D UNet. We just used this name. And HBM, just recently I discussed with some people, we have presented that at HBM, this tiramisu net. And there were some people at MIT who also just were really amazed that we already had this running, because it's really the latest state of the art of deep learning. But the name is from a new architecture, which is an advancement of UNets.
Yes. Do we have more questions from the audience? I have another question from Marius. In case you have followed the talk of Uri Hasson, he introduced this technique of intersubject correlation, and along a paradigm of making decisions, right, using language. And predicting the kind of next word. So which one is the next word? And some words were very easy to predict, and they got it 100% right. And other words were very difficult to predict. So now, seeing those talks next to each other, I really thought, a nice paradigm would be to take your EEG-fMRI combination, combine that with Uri's approach when you have the decision to make. It's a decision which word comes next, to deliberate. If it's very easy, it's going to be quick, and if it's very difficult, it might take longer. So I was to the question, I was wondering whether you have ever considered to use this intersubject correlation, which is a totally different approach in some respect. If you see a version of combining these kind of approaches of language and decision making.
Right. So actually, that technique has been applied to EEG data as well, albeit in a different context, primarily to identify when brains actually sync, right? When they're watching some kind of naturalistic video. And I think it was in the context of neuromarketing, in fact, watching video clips and trying to identify sections of those clips where brains actually kind of sync and correlate quite reliably to identify the most relevant or the most exciting bits of the commercial or something to that extent. And that was done exclusively with EEG. So it could be another way of, again, you would need a specific kind of paradigm, I suppose, that you want to tap into. But it's one of the many approaches one can take to enable the fusion between EEG and fMRI. The output of that approach can potentially be used to look at fMRI data as well and identify those signatures that are in that particular scenario most consistent across participants. So in fact, that can link back to the earlier question where, looking potentially for replicability across brains and that sort of thing. In terms of making predictions, decision-making in language in particular, I'll have to think about that, but it's certainly one other way to go about looking at the data and fusing the two modalities, for sure.
Welcome, Matthew, to the Q&A session. We had decided to start right after the last talk, and since the talks didn't follow the schedule by just run through the breaks, we start a little bit earlier, and there might be some audience people coming in later, but I'm not sure which schedule they follow, but currently we have a full room, and so welcome. And Lucy, there's another question.
I don't see this.
Okay. Matthew, I had a question for you. I don't remember, we might have discussed that earlier, but since you're showing layer 2-3 cells in human, and of course in the rodent work you're referring to the layer 5 pyramidal cells, is it technically possible to also record layer 5 pyramidal cells in human? And is that something you're already working at, or are there technical problems?
It is that there's actually a paper that preceded ours from Mark Harnett's group. I'm just trying to think what journal it came out in. I think it's Neuron. And they found some really interesting things in vitro. Basically, they found also the same uncoupled state, and they also found that there weren't the big, or at least as well sustained plateau potentials in the distal dendrite, which was surprising. And all in all, it looks like on the face of the in vitro data from layer 5 cells that you would predict at the moment that there would be almost no influence of tuft input to the human layer 5 cells. But in the meantime, we were doing the study on the linking of the two compartments that changes dramatically between anesthetized and awake rodents, and we've been studying the in vitro results, the experiments that relate to that in rodents, and here we assume that the in vitro situation is more like the anesthetized state, at least with respect to this particular mechanism, which is in the anesthetized state you have a lack of thalamocortical input to the appropriate receptors to link the two compartments, and we assume that's true in vitro, that there'll be a lack of thalamocortical inputs because there's no thalamocortical inputs at all. They've been cut away, and so we do the inverse experiment, which is to use an agonist to stimulate the receptors that the thalamocortical inputs would otherwise have connected, and we can restore coupling in a slice between the two compartments. So one possibility, which I think is a strong possibility, is that that in the human tissue it works the same way, and that because they're more electrically uncoupled, it appears at first glance that there's really no influence of the distal compartment with the proximal compartment, but that the corollary of that statement is that you would now expect there to be a more dramatic influence of thalamocortical input on the receptors that do couple these two regions, and then we're very interested now to do exactly the experiments you're suggesting in vitro, and see whether or not they then become coupled. You're asking, is it technically more difficult? It is more. Certain aspects of it are more difficult and certain aspects are easier. It's more difficult to get a lump of tissue from a patient which doesn't come with any particular orientation or clues as to where how the neurons lie in that tissue, and to get it right to do a slice where you don't damage the dendrites, and that becomes harder and harder the longer and longer the neuron is. So yeah, that's a difficulty that's hard to solve. Albert's getting better and better at this, and I'm not sure if we're doing any better than any other group at this point on that. There's several groups trying to solve this, but on the other hand, the dendrite is just massive in the layer 5 human neuron, so it's just about like patching a cell body in terms of the difficulty level once you know where it is and so on. So actually, there's it'll be interesting to see how far we get along that path. Albert's decided to stay on longer, so we have I think good cards to solve this problem, and I think it would be, well, whichever way it comes out, it'll be really exciting to see whether or not we can recreate an awake state in human tissue, and then see how the neuron comes alive under that situation.
Thank you. Yeah, exciting to see that come along. But the one idea is that the kind of integration mechanism will always be contextualizing the feed-forward input. So it could be that it's a two-compartment integration is a valid simplification of the very complicated architecture.
Well, yeah, I think that. So we struggle with this all the time. Basically, best way to encapsulate the neuron, and whether one should reduce it to two compartments and just call the rest a complication, or whether you really can't understand the neuron and the computation going on unless you have multiple compartments. Right at the moment, because of the stuff from the anesthesia, we're going back to seeing the neuron with three compartments in its basic form, and to call the coupling compartment a compartment in itself. And I think computationally you don't necessarily have to do this, but from a physiological standpoint, I think it's becoming clear that there are events going on in the coupling compartment and receptors that are there that have to be accounted for, and therefore, and also inputs that are very specific, coming from subcortical regions to that region. So although in principle you could abstract all that away by talking about how coupled the other two compartments are, but for instance, if there's some plasticity at that compartment on top of all of the inputs and the receptors and the activity itself in that region, but you want to somehow capture how this changes, and maybe this would make it a perfect kind of attentional mechanism that would feed into perhaps something related to memory, then you may want to keep the compartment so you can deal with that. I was really interested in look, I just finished watching Matthew's talk, so I only just got through the session, but so I was interested to see the Funkerkler results showing that memory, there's a residual memory in the outer layers and not particularly in the middle layers, which is something that, well, we've been looking more at layer 1 for memory in more recent experiments. I didn't talk about that in this, but it'd be really interesting to get together and talk about your results on.
Yeah, for sure. I think there's an interesting debate there, which is, so in a lot of the studies where we haven't used a visual input, or there's just been, for instance, a gray hole or a gray circle on the screen, then there's no feed-forward drive. So you're not stimulating layer 4 in any way, but you are able to drive spiking across the cortical column with purely contextual input. So it's interesting to think how that's going to be set up in the cortical circuit, and how you can actually use purely what we normally think of as modulatory inputs to actually drive spiking across the cortical circuit. That's quite an interesting future area to explore.
Yeah, and now that the resolution seems to be getting better and better, both spatially and temporally,
yeah,
and we've got a chance to look at this.
Lucy?
Yeah. Oh, yeah, we have many questions now coming in, so thank you. I'll just go through them in order. So the next one from Matteo for Marios, please. So I was wondering how you deal with subcortical structures using EEG-informed fMRI. In your work, you showed 2 different stages of thalamus processing. Have you associated an entire functional network with features extracted from EEG? In some cases, subcortical structures tend to process stimuli faster than cortical areas. So how can you be sure that cortical structures process stimuli at the same time as cortical areas?
Yeah, I think that's a great question. I think one thing to clarify for everyone is that obviously these subcortical structures don't contribute strongly, or maybe not even at all, to the cortical signals we record with EEG. So the way we would expose them in an EEG-informed fMRI analysis would be by virtue of the fact that they happen to co-vary on a trial-by-trial basis with the cortical sources of our EEG activity. So we get to them sort of indirectly through that approach. So indeed, they could process things a little faster than the cortical structures that were used to expose them. But I think the important take-home message, especially in the example that I provided in the talk, was that we identified two separate systems, an early and a late one, that were associated with different functional roles. And then the thalamus was part of that earlier system. So in that respect, you know, the relative timing between the early and the late system is what effectively was one of the main messages there. Now, of course, yeah, how does the thalamus, what's the timing of the thalamus within that early system? That's a good question. One thing we could do, and we typically follow all this up, once we have decoupled the networks in the way we did, we look at some subsequent connectivity analysis, not only across the early and the late systems in that example, but also within the individual systems. Again, that won't necessarily give you a timing, but given the direct, you know, the directionality of the effects that we're exposed to, let's say something like DCM, can give a clue about the relative timing of things within that earlier system, thalamus coming online before some of the cortical structures. Again, not an exact timing per se, but relative timing within the system. And of course, we have collaborations with people doing invasive recordings in humans during DBS procedures, for instance. And of course, this work can, you know, they tap into specific areas, often subcortical structures. So, and I view this work as a way of making more informed decisions on how to design and ultimately exploit some of those more invasive experiments that we put forward, and make more informed sort of hypotheses and questions to look at. Yep.
Fantastic. Thank you, Mario. So we now have a question for Rainer, please. So do you think it's possible to extend Sendin's 2019 work to reconstruct natural images? So which are the limitations, so acquisition, method, so forth, to reach a full image reconstruction from fMRI signals, if you think it's possible?
Yeah, that's a very good question, and we try to push the limits there. First, from the problem itself, the top-down signals, when you imagine, in our case, letter shapes, are really weak, I have to say. So in my presentation, I showed that we use denoising deep learning networks to improve, to kind of bring the noisy top-down patterns closer to the ones we have measured during perception. And if we get a bit closer, the reconstructions are much better. So therefore, at the moment, giving the noise, I'm not so optimistic that it's kind of easy to extend that to, say, complex natural images. What, however, works, which is, I think, quite surprising, and we just presented that at OHVM meeting, that we can do this all in real time. So we have really shown now that we have mathematically built new PRF decoding techniques which work in real time. So you don't need to run it over some time, but you get it really all in real time. And then we get, after about 5 seconds, images which we now show directly in real time. That means 5 seconds later to the subject in the scanner. And the person, if the letter, in our case, it's a letter shapes, is not exactly what he wanted to convey, he can continue imagining partial parts of that letter to enhance things. So that the discriminability with some kind of mixture of top-down imagery plus working on my own mental image in front of me now can make it possible to make more complex shapes, actually. You kind of paint in front of you, and that starts to work. I'm not saying it's all perfect, but we see that it's possible. So our goal is sticking to letters for now, because we want to have the possibility that people imagine all 26 letters of the English alphabet to spell out letters and words for kind of locked-in patients for communication, at least in principle. And we also have contact to Liège, where people have also a 7T scanner now, and they have locked-in patients. So we really plan maybe in a year or so to really run this kind of work on real patients, which could then really using their imagery of the letters to communicate. But of course, the core question relates also to that, because it would be much nicer if these persons, for example, patients imagine a word directly or an image, a scene, which so a glass of beer, which they just don't have to spell, but just imagine. That would be, of course, the holy grail. We are not sure how far we will go, but this will be something we will explore. The problem is that the resolution, even we have 0.8 millimeter voxels, do not allow now to get the featural representations in V1. So you get the topography, and you basically only use that in a noisy way to reconstruct the images. I mean, in the talk of Andrew, you heard the so-called line scanning technique. That is a wonderful technique because it really gives you the resolution we would need to potentially reconstruct more complex features like orientation, tuning, and so on. We could probably do much better reconstructions. The problem is that, again, loses coverage. You have then only a very small part of the brain. And in our case, we do the opposite. We have large coverage, so the full visual cortex, and we use the topographic patterns to kind of decode the imagined top-down information. Last point, we also now, actually using the tiramisu network I presented, remodel the whole data and doing it at a laminar reconstruction. And it seems that if we use only the upper layers, which we haven't done in the published paper, the reconstruction gets sharper. So it at least gives some boost in quality, which again, we want to exploit in the future, where we again benefit from laminar fMRI.
Thanks, Rainer. I have a follow-up question for you, actually, from Renzo. The tiramisu is fantastic. I would like to use it on my data. How specific is it trained to a specific MRI protocol? So can we use it with slightly unconventional T1 weighted EPI data?
That's a very good question. Actually, unfortunately not. We have found out with Marian Schneider, who did the main work on this, that it is quite sensitive to the specific sequence we use. So therefore, we have now, and we make this all publicly available, we have actually two trained networks. One is based on an MP range sequence and one on MP2 range sequence without any pre-processing. So I know that Renzo post-processes, for example, MP2 range sequences to get rid of the background noise. This network we have trained doesn't need that. You start with completely noisy images as they come from the scanner, from the standard MP2 range sequence or MP range sequence. But we use then two trained networks with very limited training data. But then from this kind of sequence, we get to kind of any data. I'm testing now a lot of brains, very, very, very good results. But it is sensitive because we use only about n equals 5-4 brains. We basically are somewhat dependent on the specifics of the scanner sequence used. The good thing is, since we need so little training data, we could use some of Renzo's EPIs, high resolution EPIs, and if he has segmented them already, we could train with only about 5. That's, I think, is the big advantage of this tiramisu net. The training data is very limited, and you get very good generalization, outperforming other architectures of deep learning. And therefore, I think that would be a great chance, and that's what we want to do now. We want to apply it to also functional data, to segment it, and so on. And actually, it would be a nice collaboration with Renzo, who is around the corner, to do this for functional data next.
Thanks, Rainer. So you mentioned line scanning there as well. So Tyler, we have a question for you, also from Renzo. So the line scanning is awesome. 200 micrometers over 100 milliseconds is mesmerizing. Would you say that you superseded the resolution of the hemodynamic response? Or is the neurovascular coupling even more specific than that?
Yeah, that's a really, really good question. And I think it's an active area of research that hopefully we can contribute to. I know that Laura Lewis's lab is also looking at very high temporal resolution scanning, although with a different technique. And interestingly, they've been finding that the hemodynamic response actually gets more specific when using really fast stimuli, which was something that we did with our movie stimulus. So I think it needs to be a combination of both the acquisition technique as well as the experimental technique. But absolutely, obviously 100 milliseconds is faster than the response function that we're looking at. But I really hope to see progress in this area. I think it's a really interesting area of research that potentially could answer some really interesting questions, specifically when comparing against other modalities like both EEG as well as intracranial recordings in both humans and in animals.
May I add a short answer to that, a short point to that? We have also experimented with a sequence coming from Jürgen Hennig's group, MR physics guy, which is called MREG in our lab. And it has 100 millisecond resolution and basically whole brain coverage. Of course, not the spatial resolution of line scanning, but we see the same thing. We see amazingly temporal, so you see not only the physiology, you see the pulsation, for example, in your time course and so on, but you also need much more sophisticated models for serial correlation correction. So an AR1, AR2 model is insufficient. You need to capture the high temporal resolution serial correlations. And if you remove that, there are still temporal effects, which I think really will be important to investigate more in the future. So I confirm with another sequence with similar temporal resolution that this is indeed something too much unexplored in fMRI, because there might be some very important jewels to dig out.
Thank you, Rainer. So we have a question now for Matthew Larkum. So are there any proposed mechanisms for how neurons distinguish OR and XOR inputs into the dendritic tree on an ion channel or receptor level?
Yeah, do you mind if I share my screen for answering that?
Yeah.
Let's see. Do you see that? Okay. So yes, we think that what we saw in the human layer 2-3 neurons was a receptor, well, a channel that that inactivates with increasing stimulus, so that there's a sudden, there's still a threshold for opening this channel that appears to be a calcium channel, and that at increasing strength it inactivates again. So it's as it says here, sharply tuned, and we saw that that's basically gives the compartment a particular activation function. And the normal, in the normal case, you would simply get, well, at most channels would inactivate at some intensities, but not realistic intensities. So most of the time you would get only more response to increasing stimulus. But then if you, let me just, if I go to another one on that same presentation, here we go. Yeah. So if you then consider that the neuron is a complex network of different compartments, and the different compartments might have different channels with different activation functions. Some of them may be sigmoidal like this, and now none of these are drawn with this particular activation function that we've just found. But if you now insert, instead of a sigmoid, into some of these compartments inactivation functions, then first of all, the different parts of the neuron might be responding differently to, let's say, have different rules for how they compute inputs. but it would be far more complex than that in the larger scheme of things when you start mixing the influences of the different compartments on each other. And so in a sense, you have a mini network of logic gates, or you have the possibility for a network of logic gates, and therefore really complex functionality that the neuron could compute, given that one compartment might be inactivating with certain connections. One compartment might be doing an inclusive OR, and then they might be interacting with each other. And they're interacting with each other not just in an instant in time, but over time as well. So that there'd be the possibility for some kind of memory function within the neuron itself. So the possible complexity of a neuron, from the standpoint of where we are at the moment, knows no bounds. But what encapsulates its actual function, and to what extent these possibilities are instantiated in the actual operation of the brain is really unclear at this point. But well, I guess that means there's a lot of exciting stuff to come.
Thank you, Matthew. Fantastic. So I think we have another question, a more general question for everyone. So it's a question about the generalization of the knowledge presented here, benchmarking, interpretation, and so forth, about layer fMRI in early visual areas to other cortical regions. So to what extent could this be transposed to higher level cortical regions, such as prefrontal, parietal, or to other sensory cortices?
Yeah, maybe I can start on that one. So we see obviously this big difference between a feed forward profile in the bold and a feedback profile. We get this V-shaped profile for feedback effects. And the question could be, does that translate to other areas? So, and I think the answer to that is probably going to be, it's going to translate to primary sensory areas, the primary auditory cortex, for instance. But it's unlikely to be translating to something like primary motor cortex. So we know, of course, there is some studies, laminar EFIS studies in, for instance, V4 by John Reynolds' group, that have already shown that if you look at attentional top-down effects in V4, these are strongest actually in layer 4. So it's quite a different profile to what we've seen in V1, where they're weakest in layer 4. So the exact shape of these top-down or cognitive effects that you see are probably going to vary as you move up the hierarchy. And if you get to something like primary motor cortex, which doesn't even have a layer 4, then of course you're going to get quite a different sort of profile there. I think what will be generalizable is the ability to take laminar EFIS studies, look at the pattern of spiking across the layers, and then use that as a prediction for what you're going to see in MRI. So I think you will be able to still be able to benchmark or still be able to use the animal studies as a guide for what you're going to see in fMRI studies and stuff. So we'd hope that would translate.
Can I continue? Actually, it's of course very good if we can ground the human findings on such EFIS experiments also in higher cortex. But sometimes that is not yet available. But there is at least one or a couple of papers which have shown laminar fMRI already in prefrontal cortex. Not only motor cortex, really prefrontal cortex. For example, a group of NIH, I forgot the first author, but Renzo Huber is also involved, who asked a question earlier, where they showed that if you separate working memory tasks between different modes, like more rehearsal mode, more manipulation mode, you find very consistent laminar changes across depths with fMRI kind of telling you which of the tasks, of these two tasks, the person is basically performing. So therefore, at least this shows that the usefulness of laminar fMRI seems in the human brain also be possible to do in cognitive domains and higher level cortex, like prefrontal cortex. So therefore, I think that there's not a strict limitation. But of course, to know exactly what we see, somehow we always feel we need to show that it makes sense with respect to what you see, say, in the macaque monkey. And there, if you have trained experiments, they differ to some extent, of course. The more cognitive you go, the more there is a difference that a human can very flexibly change sensory motor mappings, for example, which in the monkey you usually train thousands of times that can perform a task. And of course, the connectivity and the network dynamics might then change also because you have so many trials performed. In the human, you can study the higher flexibility of the human cognition, this laminar fMRI, and that will be for me very exciting because you might then also go into processes which you cannot so easily do in the animal research. I'm a fan of animal research, but of course we have to also find the neural correlates of the more uniquely human capabilities.
Yeah, absolutely. I agree with that. I think the key thing, maybe the point I'm trying to make with the benchmarking studies is that it shows we can trust what's coming out of the MRI signal. So they are revealing something which is also, we can see if we look with electrodes and we actually put them in the cortex. So the more benchmarks we have, the more we can trust the actual signal that we're measuring with laminar fMRI. I think that's an important thing to have in our back pocket.
Can I add to this discussion too? Just hopping again on the perspective that Lars Muckli and I were pushing. I think it's already a remarkable fact of the cortex that there's this kind of uniformity to the cortical column, and in particular things like pyramidal neurons looking largely the same in one region of the cortex than the other. And I think the properties that are emerging from the pyramidal neurons in the different regions, so in primary sensory versus motor versus prefrontal, are coming out with more similarities than differences. If we want, as far as it seems at the moment, that's a fair generalization. And on the other hand, I think what Matthew is just saying is also true, that there's also clear differences in the functional responses of the different regions of the brain under different circumstances and so on. And in particular, what we don't know at this point, people are drawing these simplified diagrams of how feed forward goes this way and feedback goes that way. And I think anyone who's had to draw such a diagram is faced with that problem of labeling these inputs, because it's even feed forward and feedback are hard concepts to get consistently right for every region of the brain and so on. For instance, I mean, should we call the inputs to motor cortex feed forward or feedback, if it's right at the end of the loop or perhaps it's right at the front? I would be guessing at this point that when push comes to shove, the designation of what these inputs and outputs represent will actually closely tie to what the functional properties are of the pyramidal neuron, and that these words actually make less sense than talking about the, let's say, apical input versus basal input, and apical output versus basal output, and so on. And that when one then goes back to try to interpret, let's say, the fMRI signal coming out of the prefrontal cortex, you'll then be asking yourselves, well, what is that implying about the functional input and the functional output to this region, which clearly is quite different from region to region. That's not uniform over all the regions. But if there is let's say a uniform or a universal way to consider the way the components operate, which on face value seems to be a reasonable assumption at this point, then you would have predictions about, I guess we would start to use different terminology to describe what's going in and out of the components that are universal. That would give us better and easier discussions, let's say, about how we interpret what prefrontal is doing and what primary sensory is doing, and so on and so forth. So, but at this point, I would guess we're in paradigm-changing region of or time of neuroscience in which the terminologies confuse us in terms of how to actually describe what's going on in the different regions. That's my take on it at the moment. I think Lars would largely agree with that.
Yes, I mean, I wonder. You showed in the introduction the evolutionary elaborated intermediate layers of the superficial cortex in humans compared to monkeys and rodents. So I wonder if we will learn more about the capabilities of contextualizing that humans have, maybe better than monkeys or rodents. And that might currently be a limit of fMRI, because the separation of these superficial layers will not be possible. I think we have one more question.
Oh, could I just
yes,
just address that, because I think that also ties to what Rainer was saying, that I think the latest results that we were getting from human tissue is a cautionary tale not to generalize too much from rodents and to just assume that everything we've seen in a rodent will extrapolate to human tissue, and particularly if we're going to start talking about universal properties of a cortical column and the components of them, and then you suddenly find that there are interesting capabilities of human neurons versus rodent neurons. Then of course the same project that I'm talking about becomes specialized on a species basis as well, and then you would have to reformulate what the universal function of the cortical column or of any given component in the cortical column should be described as, and then reapply as necessary to the human data, which could come out quite differently in the end.
Thanks, Matthew. So we have another question from Narendra. So it's a question for Matthew self, please. So really excellent talk, thank you. Did you find any features of the fMRI data that were inconsistent with what was measured physiologically?
Right, well, there are some issues which come down to analysis techniques. So if you look at gradient echo bold, for instance, if you just take the raw laminar profile of that and you don't allow for the fact that it increases towards the superficial layers because of these draining veins on the superficial layers, then it's not going to match up with the underlying electrophysiological signal. So you're going to get a mismatch there. But if you use techniques like decoding techniques, or look at tuning strengths, or you mask out the draining veins, things like that, then that brings it back closer to what we see in electrophysiology. One thing we probably consistently see throughout our studies is that the MRI signal seems to be perhaps more sensitive to modulatory or feedback inputs than the spiking activity. So it's more exaggerated in the MRI signal than it is in the EFIS signals. So for some things, for instance, like top-down modulation through attention, at the spiking level, they can be quite subtle. They can be quite subtle modulations of spiking activity. If you look in the bold signal, they can be quite large modulations of activity. So maybe because of the way that the MRI signal is being generated, but how the bold signal is being generated, it's more sensitive to these modulatory kind of top-down inputs. And it might actually be more sensitive tool than EFIS to actually reveal some of these modulatory inputs. That seems to be a difference.
Well, thank you very much. Thanks for the speakers. Thanks for the audience. If there are no further urgent question, then we'll come to a close. I would like to also mention that there is a further booth from the Human Brain Project that will also engage with you in case there are questions about the capabilities of eBrains. You have seen throughout the mini conference that we have been working, and we're working towards combined papers that we call live papers, in which we will try to bridge scales and combine human fMRI across species like monkeys, but also down to rodents, to compare principles. And the data of these kind of studies will also be shared through eBrains. So that's one of the kind of advantages that we are working towards. And if you're interested in learning more about this, there is a booth, and there's more information at a landing page, which is called https Human Brain Project EU, and then EU FENS 2020. And with this, I thank the audience and especially the speakers for the brilliant talks. Thank you very much. Bye-bye.
Okay.